Hi everyone, so this is a second lecture on the techniques in microbiology. So we will learn about isolation and cultivation of microorganisms. We will particularly learn about isolation and cultivation of bacteria. Okay, media is a substance which is used by the bacteria to grow. So the media can be divided into liquid and agar media. So liquid media also called as a broth, so which is present in a liquid form. On the other hand, agar media is prepared the same way as a liquid media, except agar is added. So agar is a gelling agent. So after you added the agar, so we plate it, or you can also put it in a test tube. Okay, first is the culture. So culture is an organism that grows on or in the media. So the culture can be divided into a mixed culture or a pure culture. So mixed culture contains more than one kind of a microorganism, while on the other hand, pure culture contains only one kind of a microorganism. So which means mixed culture means it contains more than one species of bacteria, while pure culture contains one species of microorganism. Yeah, the following term is isolation. So isolation is a process of isolating organism from a mixed culture. So which means you try to get a pure culture from a mixed culture. So the second term is isolate. So isolate is an organism that has been successfully isolated. And then the preparation of pure culture involves isolation of organisms and maintenance of the isolated colony. So whichever colony that been isolated from a mixed colony, we call as a isolate. On the other hand, the technique we need to know when it comes to isolation is the aseptic technique. So aseptic technique is a proper technique of handling apparatus, organism with the aim of to reduce contamination, and is very important technique in microbiology, which you will also learn in your practical one. Okay, isolation. Okay, look at this plate. So this plate, it contains two different types of bacteria. So the red ones are the Serratia and the blue ones are the E. coli. And then we try to isolate single colonies or isolates from the mixed culture from the process called isolation. And the same thing happens here. So we try to isolate Serratia from a mixed culture, and all of these things we call as isolate, and they are pure culture. So once you obtain this pure culture, you use them to make a stock culture. Stock culture is the culture that usually we keep it for years as a stock. Okay, let's see how pure culture can be prepared. So the technique we're going to use for the preparation of pure culture is called as a streaking. So the objective of streaking is to obtain isolated colonies which can be used to prepare the pure cultures. And the method we're going to use is the quadrant method. There's a video here which will show you like how to do the streaking. Let's watch the video together.
Place the inverted plate close to the Bunsen burner to reduce chances of contamination. Turn on the Bunsen burner. Loosen the cap of the bottle containing the inoculum. Hold an inoculation loop in your right hand and put the flame of the Bunsen burner to blue, and take the inoculating loop and heat at the peak of the blue flame until the wire is white hot, and then allowed to cool or keeping close to the Bunsen burner. Do not wave around the loop to cool quicker, as it increases the chance of contamination. Without putting the loop down, lift the test tube containing the inoculum with your left hand and remove the cap of the bottle with a little finger on your right hand. Pass the neck of the bottle through the flame 2 to 3 times so it becomes sterile. Once the bottle is sterile, put the loop into the suspension without touching the sides of the bottle. Remove the loop and reflame the neck of the bottle before closing the lid to sterilize. Partially lift the lid of the petri dish, and with the loop containing the bacteria, rub a small circle immediately into the agar on the first dot at 12 o'clock to prevent your liquid from running. Reflame the loop and let it cool for 5 seconds by touching an unused part of the agar surface close to the edge of the plate. Use the loop to streak the bacteria across a quarter of the agar from the circle you just did in 3 to 4 lines. Reflame the loop and allow to cool. Then from the area you just streak another 3 to 4 lines from 3 to 6 o'clock. Reflame the loop and then do another 3 to 4 lines across the petri dish from where you just left. Reflame the loop. In the last quarter of the agar dish, you need to streak the bacterium in a zigzag formation from the outside of the agar towards the centre. Be careful not to streak the bacterium where you previously streaked, as it will not get the desired separation, and the zigzag of the bacterium should not touch the sides of the petri dish. Reflame the loop to sterilise before putting away. Turn Bunsen burner flame back to safety, and please leave your agar plates on the side to be collected for incubation.
Okay, I hope the video is clear, like how to do this tricking. Okay, this is another slide. It's showing like how to make a pure culture using a tricking method. So let's say you've been given a mixed culture that containing Serratia and E. coli. So first, you use an inoculum loop. You transfer a little bit of the liquid into the plate in a circular motion. After that, you heat up the loop to sterilize it and then cool it down and then do 3 to 4 streakings. It's more like 3 to 4 lines using the inoculum loop. After that, you heat up the loop and then wait for it to cool down, and then from the quadrant 1, which is this one, and then you do another 3 to 4 lines. That will become your quadrant 2, and then heat up the loop, and after it's cooled, and then you do another 3 more lines from the second quadrant, and then do the following, I do the same thing for the fourth quadrant, and then followed by the zigzag. Okay, so usually after 24 hours of incubation, you should able to get a single colony at the zigzag or at the fourth quadrant. Okay, usually one of the way to certain that the culture that you are obtaining is a pure culture is that all the bacteria or all the colonies will have a similar morphology. And once you know all of them are having a similar morphology and a pure culture, and then we start to do a stock culture. So how to do a stock culture? So we will be looking into it in a while. Okay, this image showing a successful streaking. So if you look at here, so this is your quadrant 1, quadrant 2, quadrant 3, quadrant 4, and this is supposed to be your zigzag. So usually you should able to see a single isolated colony at the quadrant 4 here, and then the zigzag. So the following lectures will be on maintaining and preserving pure culture. So why we have to maintain and preserve a pure culture? Now, the reason to maintain and preserve the pure culture is to have a sustainable supply of pure cultures, either is for industrial purposes or for research. So there is various ways you can maintain and preserve a pure culture. So let's look at one by one. Okay, the first method is the agar slope cultures. So the agar slope culture is applicable to a wide range of organisms. So usually the organisms maintain on the surface of agar slope. So usually the agar slope will be formed on the test tube or a screw cap tubes. So this is a screw cap tubes and this is the test tube. So usually the agar will be formed in a slant position and the bacteria will be streaked on the surface of the slant. And then these test tubes or screw cap tubes can be stored at room temperature or in the fridge. And the requirement of subculturing depends on the species of bacteria. Let's say, for example, fast growing bacteria, the subculturing will be done every 2 weeks. But then for the slow growing bacteria, usually be done every 6 months. Okay, there's been various drawbacks of agar slope cultures has been reported for maintaining and preserving pure cultures. So agar slope cultures has been reported can lead to contamination during subculturing, and also has been reported can cause loss of viability due to faulty media. And apart from them, dehydration is another common issue or problem with the agar slope cultures due to imperfection in the seal of the container. And the loss of stability of character in the bacteria also has been widely reported for agar slope cultures. Loss of stability of character refers to the changes in the phenotypic as well as the genotypic of the bacteria. Okay, some researchers also using agar plate cultures to maintain and preserve bacteria, but it is highly inadvisable, and the main reason is that contamination can easily occur in the agar plate. Okay, some researchers are also using liquid cultures to maintain and preserve the pure cultures, but then again, this is also not encouraged because the detection of contamination is not easy in the liquid culture. Okay, some researchers also using a special media for maintaining and preservation of pure cultures. So for example, Robertson cooked meat medium. So actually this media is containing beef. Okay, this is actually the beef pellets, and usually is used for the anaerobic organisms. So anaerobic organism mean a bacteria that doesn't require oxygen for the growth, and usually the subculture is done at intervals of up to a year. So the following method is preservation under oil. So which means literally you're going to use the oil to preserve and maintain the pure cultures. So usually the oil we use is a sterile liquid paraffin or sterile mineral oil, and the culture is usually viable for several years without subculturing. So look at the pictures here. So this is the bacteria culture and this is the oil. Okay, the following method of preservation is freeze drying, or also known as a lyophilization. So usually the culture is frozen at a very low temperature and placed under a high vacuum. So when you put a liquid culture in a low temperature and a high vacuum, usually the liquid will turn into a powder. So in the powder form, the microbial growth is precluded, but the viability is maintained. So which means the bacteria usually in a dormant state, and the cultures stored in a sealed glass ampoules under vacuum. So it usually look something like this. Okay, there are few advantages of using a freeze drying for preserving pure cultures. So first of all, it easily can be stored for long periods of time. So usually the ampoules, which has been shown in the previous slide, can be stored up to like 15 to 20 years, and usually can be kept in room temperature or in the fridge. And this is particularly very useful for storage of culture collections. So culture collection here means it's more like you're making your own library of bacteria. And then this freeze drying is also very important in dispatch of cultures, because all the transport of cultures from one country to another usually is done in a freeze drying method. So the final way of preserving culture is storage in liquid nitrogen, and it is also one of the most optimal and universal method being used in preserving cultures. So it is suitable for fungi, bacteriophages, viruses, algae, protozoa, bacteria, yeast, mammalian cell and tissue cultures. So this means it can be used for any cells including eukaryotic cells. And then usually the organism suspended in a supportive medium, for example like 20% glycerol as a carbon source, and cooled at a rate of 1 degree Celsius per minute to a final temperature of minus 196. And then usually this is how the storage cabinet will look like. So this whole thing will be filled up with liquid nitrogen, and then inside you will see some boxes, or sometimes also comes in the cylinder form, so where you can arrange your bacteria in the shelves here, and then you keep it in the liquid nitrogen. Okay, one of the disadvantages of storing in liquid nitrogen is that liquid nitrogen easily evaporates, and therefore it must be replenished regularly, and this increases the preservation cost of the pure cultures. So that's all for today. If you have any question, please post in the e-learning.